Thank you so much for having me. I'm so excited to be here. I will acknowledge I am on my way to another meeting, and I was supposed to fly last night so I could be on the ground in my hotel room before I presented to you. And as with many things with flights, that didn't work. So I'm in the Atlanta airport. So everyone around me is going to learn about ovarian cancer, whether they want to or not. So hopefully that's okay. But let me share my slides. Let's see if I can share. Yep. Okay. And hopefully the background noise isn't too bad, but let me know if it gets.
We won't even notice. You just go ahead and with your slides. We're going to learn a lot. Thank you.
All right, here we go.
And we see them.
You see them? Let me get them in. So, okay. All right, so I'm going to take us through sort of where we are and where we're going very rapidly in the field of ovarian cancer. And I'm just at the beginning and going to acknowledge I'm going to be speaking primarily around high-grade serous and high-grade endometrioid ovarian cancer, but perhaps in the panel, if we had questions about rare tumors, we can discuss those as well. My objectives for today are really to kind of take us through, like I said, where we've been. Traditional chemotherapy. Chemotherapy is kind of the hungry caterpillar. Eats whatever, pretty indiscriminate, kills cancer, kills other things. Where we've been, kind of entering the chrysalis. We've tried a lot of things. They didn't work. We went back into the goo, came back out with, I think, strategies now that are going to help us all emerge and help our patients fly for longer and better lives. And I hope to convince you of that by the end of this talk. This is just a little timeline of where we have been. There we go. Where we've been, I've been a GYN oncologist since 2004. So this timeline kind of encapsulates, my career. And when I started this, we had chemo, paclitaxel and carboplatin. That's what we had. So we used the hungry caterpillar and we used it again and again and again until, it didn't work. We had a little bit of a bump. We learned some things about anti-angiogenesis, which is how we target how tumors make blood vessels for themselves. And that led to the medicine bevacizumab. You probably know it as Avastin. That we use a lot, not transformative, but pretty good. And we entered probably one of our most transformative eras for some people with ovarian cancer in 2018 and beyond, with the development and incorporation of PARP inhibitors into the treatment paradigm for our patients with ovarian cancer. So just to remind you, I'm going to kind of show you, I'm not going to show you too many of these curves today, but just want to explain a little bit of what we look at when we're looking at clinical trial results. This is the Avastin clinical trial. And this is what's called a Kaplan-Meier curve. And what this shows us is, you can see the months that are labeled here. Let me get my little cursor up going here. The months that are here. So these are the medians. And you'll hear people a lot of times cite medians, the median survival, the median progression-free survival. I don't like to talk in medians. Median's 1 point on a curve. It's an average. And it doesn't represent any person out there. It's not a label for you. So I don't like to talk in medians. What we like to talk about is the reduction in risk that a certain intervention, in this case Avastin, brings to a therapy. So Avastin, you can think, if you add Avastin to frontline chemotherapy, it reduces the rate at which progression occurs by about 38%. And so you can say, well, that sounds really good, or you could say that sounds modest. That's a judgment call. But that's sort of the expectation, whatever a patient's trajectory was going to be on paclitaxel and carboplatin alone, the addition of bevacizumab will slow the rate of progression for that person, wherever they were, by 38%. And so that amount is going to differ for each individual person. And that's how we think about these Kaplan-Meier curves. Medians are a really big misrepresentation, and they scare people, actually. When we say median survival is this, they think that, well, that means that's my expiration date. And nothing could be farther from the truth. So I always like to explain that to patients, because it's kind of scary to see these curves and try to interpret them without understanding that. So that's the Avastin era. Let's talk about PARP inhibitors. PARP inhibitors right now are approved in the United States for patients whose tumors are caused by a BRCA mutation, or whose tumors harbor some deficiency in how they repair the DNA that is caused. that is the DNA damage that's caused by chemotherapy. And that's called homologous recombination deficiency, or HRD. Those are the two biomarkers in which we can use PARP inhibitors for maintenance therapy after we give chemotherapy. That's about 45-ish% of patients who are newly diagnosed with high-grade serous or high-grade endometrioid ovarian cancer. Where did that data come from? I'm not going to show you all the studies. I'm going to show you two representative studies. One is SOLO-1, which brought olaparib, or you might know it as Lynparza, into use in patients with BRCA whose tumor responds to chemo, which most do, and then they were randomized to get 2 years of olaparib or placebo. And I'll remind you, at the time this was done, the standard of care was placebo, was nothing. We just monitored. So this seems today to be almost unethical, but 12 years ago, this was the standard of care. And this is the Kaplan-Meier curve. So remember, I told you how to interpret these. So what we can say is in patients with BRCA who respond to paclitaxel and carboplatin, use of olaparib reduced the rate at which progression occurs by 70%. And in many cases, progression has never occurred. And so we can say with some assuredness, although we have not reached final analysis for SOLO yet, and we won't for many years because people are doing so well, we do think that there is a fraction of patients who have been moved into that cure fraction by use of olaparib in this biomarker selected group. The absolute percentage of cure, we can't say yet. At 8 years, we're sitting at about 45% of patients who enrolled in this clinical trial, but we'll get updated numbers on that as they come. But that compares to 20%. And when I say cure, what I mean is patients were treated with chemo and a PARP, and their tumor never came back. That's different than overall survival, which is maybe it came back and we treated it with effective therapies and we keep on treating it. That's overall survival. That's a little different than disease-free survival, which is our metric of cure. And just to kind of put this in a little more of a hopeful frame, we've never talked about cure before in ovarian cancer. It's just not something we've been able to really talk about with rare exception. And now we are actually talking about it, and it's sort of become a reasonable goal to strive for finally, because we have an emerging set of novel therapies that may really impact on us. But I think there's a lot of reasons to be optimistic as I take you through this. So that's just BRCA. That's about 20% of ovarian cancer. What about everyone else? Well, this is the PRIMA study. Similar, but a little different. PRIMA used the PARP inhibitor niraparib, and it included everybody. It didn't have to have any biomarker, and the only requirement was that patients' tumors had responded to paclitaxel and carboplatin, and then they were randomized, same, just like SOLO, to receive a PARP inhibitor, niraparib, which you guys know is Zejula, or placebo. And then they looked at it by the different biomarkers. And so this is the biomarker group of tumors that are homologous recombination repair deficient. It includes BRCA, but it also includes about 25% more people whose tumors were not BRCA, but were homologous recombination deficient. And you can see here that for patients who received niraparib, the reduction in the rate of progression was about 50%, 57%. So that's pretty striking. And we do think that in this biomarker selected population, we're still following. We also may have cures. We're still following. So we can't say it yet, but we are hopeful that in this non-BRCA, but still HRD positive tumor type, we'll see more patients transformed into cures. So these PARP inhibitors have been transformative in these two biomarker selected groups. And this is just a summary of kind of, just so I'm fair and balanced. There's PAOLA-1, which was olaparib, PRIMA, niraparib, and then Athena was rucaparib, all very consistent. There's really no difference in the PARP inhibitors, very consistent improvement in reducing the rate of progression. And we're now trying to really hone in on how much PARP does someone really need? And this is an ongoing clinical trial trying to ask the question, do you really need 2 years of PARP inhibitor? Maybe you just need 1 year of PARP inhibitor. So trying to get at quality of life and not exposing people to too much medication if they don't need it, which is a very important question. I will tell you, we came up with 2 years a little bit arbitrarily because I was involved in designing Solo One. Do we need 2? I don't know. We might just need 1. I would much rather use 1 year if it doesn't diminish efficacy. So this study is happening right now, and we're quite excited to see whether or not we can de-escalate the amount of medicines we give to people over time. Where we have not yet transformed outcomes is in really the majority of people with ovarian cancer, and those are those tumors that are homologous recombination deficiency test negative. Here, Avastin or bevacizumab has a modest impact. And so you could say that could be a standard of care. Some people say, it's not curing anyone, so maybe we just do close monitoring because that'll be the time when people can actually enjoy some time off therapy. But this is a place where tumors don't respond as well usually, and we really need to up our game in terms of transforming outcomes for this group of patients, which is about 50%. So we have a lot of work to do, and we're already at it. And I'm going to talk about antibody drug conjugates in a little bit, but this is one of many studies coming. This is the first one in public domain, so I can show it to you. Enrolling patients whose tumors, they don't even have to have responded. They just have to be not progressing to chemo, who have a biomarker, they're HRD test negative, but they have a biomarker of HER2 expression, 1+, 2+, or 3+, which is maybe 30% of high-grade serous ovarian cancer, and randomizing for maintenance to Avastin or Avastin plus the antibody drug conjugate called trastuzumab deruxtecan. You will know the commercial name is Enhertu. So this is the first publicly available look at an antibody drug conjugate in a randomized phase 3 study. This one is an investigator-initiated trial by Dr. Rebecca Arend at Alabama, bringing the antibody drug conjugate Elahere or mirvetuximab for patients with folate receptor alpha high tumors with carboplatin and to follow as maintenance. So we're starting to see, you're going to see more of these antibody drug conjugates coming in as maintenance, trying to really transform outcomes in the front line. And I'm very excited to see, what these do. We do see recurrences though, and how do we think about recurrences? This is changing, but right now we still think about recurrences somewhat, it's like a little bit of a dinosaur. We still look at the length of time from the last platinum. So if the last platinum was more than 6 months ago, we characterize that tumor as possibly platinum sensitive, and we reuse carboplatin doublets. If it's less than 6 months, we tend to use things other than carboplatin. That definition is changing though, and I'll tell you a little bit why. This is just a table of the 3 big studies that were done that set the standard of care for platinum sensitive recurrent treatment. Carboplatin plus one of 3 other chemos. You can use paclitaxel again, you could use Doxil, you could use gemcitabine. They're all about the same. And if you add bevacizumab, there's a little bit of survival advantage. And so we like that as an option. I'll tell you though, all these studies were done in an era before we were using PARP inhibitors in frontline. None of these patients had received prior bevacizumab. So there is a wonder about whether or not these results would still be the best thing, for patients in that situation. So we're really trying to think now in a modern era, how do you take care of a patient with recurrent disease, considering more things than just the time from last platinum? And then can we reuse PARP inhibitor in that setting? Would I ever reuse a PARP inhibitor? Someone that got a PARP inhibitor before. The FDA right now says we shouldn't do that outside of a patient with a BRCA mutation, but we all wonder whether or not we should do it ever, or whether or not it's okay to do it in a patient who maybe got 2 years of PARP inhibitor and maybe recurred 3 years later and responds again to platinum. I might use PARP again there. In somebody who recurred at 18 months into their PARP inhibitor, gets a platinum and responds again, I might not use a PARP inhibitor again. So these are all of sort of the nuances that we don't have answers to yet, but we're really trying to get to with ongoing clinical trials. And we're trying to redefine how we think about you all. And we've been doing this. This isn't new. This is an old study that said, okay, if I have a patient who recurs between 6 and 12 months, that's technically platinum sensitive, but maybe platinum's not the best thing. And so they said, well, we're going to use a novel combination. In this case, it was Doxil plus trabectedin, but that's not super important. I'm just saying it didn't work. They tried to substitute something and it didn't work. This was years ago. And we kind of said, well, we shouldn't do that again. But again, I don't think Doxil and trabectedin is the best agent. We have better drugs now. So maybe just having a better drug would have made this successful. And B, these patients had progressed 6 to 12 months, but they hadn't seen a PARP inhibitor. They hadn't seen anything else. And our population has vastly changed. So we're starting to rethink these definitions and what's the best therapy to offer each individual patient based on a lot of factors, not just the platinum-free interval. So what do I mean by that? Well, we have a little bit of data coming out to sort of whet our appetites with, again, antibody drug conjugates. This is the biggest study thus far to be published using, again, the folate targeting antibody drug conjugate mirvetuximab called Elahere. And this was led by Dr. Angeles Alvarez Secord in patients who were tumors were platinum sensitive, but they'd seen at least 2 platinums. So this was their second recurrence, third recurrence, still platinum sensitive. And you can see, I think I have some boxes. In the overall population, the response rate, meaning the tumors shrank by at least 30%, that's what response rate means, was 52%. That's pretty good. In patients who'd never seen a PARP, the response rate was 75%. It's only 12 patients, so we have to take that with a grain of salt. But in patients that had prior PARP inhibitor and their tumor progressed during the PARP, the response rate was 45%. So these all seem pretty good, especially when you think that we're in 3rd or 4th line therapy. The question is, how would platinum have done in that setting? Is this better? Is it the same? Or is it worse? Until we have a control arm, we really can't answer that question. This is data on a different antibody drug conjugate called raludotatug. It doesn't have an easier name yet. This targets cadherin-6 with 18 patients with platinum-sensitive recurrent disease with a response rate of 72%, 58% amongst those tumors that have progressing apart. And we're seeing each of these antibody drug conjugates has had a small number, except for MERV, which has 80 patients, small number of patients who participated in clinical trials, and you can see the response rates are over 50%, giving us a lot of optimism that this may be a better way to come at recurrent disease rather than just retreating with platinum. But we're going to have to put it to the test in clinical trials. Versus, do you retreat with platinum? And then in that group of patients that responds, you use an antibody drug conjugate for maintenance. And these studies are running. Again, mirvetuximab being the first antibody drug conjugate approved is out in front. This study will be presented at ESMO in a few weeks, we believe. This is study 420, replacing paclitaxel with mirvetuximab and carboplatin, and then mirvetuximab maintenance. And then the Gloriosa study, which I think I have a schema here, is for patients who get platinum again and don't progress. And then they are randomized to Avastin alone or Avastin plus mirvetuximab as maintenance. This will be the first antibody drug conjugate in the maintenance setting to read out. The study just completed. I believe it closed to accrual, or it's close to closing maybe. But it'll be the first one to read out. So this will be exciting to see whether or not this moves into an earlier line of therapy. And then for tumors that we say shouldn't get platinum anymore, they're platinum resistant. This is the expectations with chemo, the hungry caterpillar. It's just not acceptable. I mean, this just doesn't justify the toxicity with response rates 10%. Why don't these work? It's chemo. Why does it kill the cancer? Well, the hungry caterpillar chemo can eat a lot of cancer, but the cancers, by the time they're resistant, they grow faster than the caterpillar can eat them, just like grass. And so they just are outnumbered, or they get eaten. by resistance mechanisms or efflux pumps, whatever chemo you're giving, tumors develop a way to just kick it out. And so it doesn't do its work. So we have to think about these things and get more targeted with our interventions. Now, is that true for all chemos? No. Weekly paclitaxel is a very effective therapy in the recurrent resistant setting, with several studies that I'm showing you here, all with response rates. I have a little chart in the 30% range, which is kind of the floor for what we think is interesting to study in a platinum-resistant setting. If it's any less than 30%, we're not interested in moving that forward. Greater than 30%, we're like, okay, there could be something here. But weekly paclitaxel meets that bar. And so we do think it is one of the most effective therapies by itself or with bevacizumab in a resistant setting. And then that's based on the Aurelia study, which is about a decade old. But that combination of weekly paclitaxel plus bevacizumab gave us all the idea that if we could modify the chemotherapy and make it a hungrier caterpillar, maybe it would work better. And that drove some of the evolution in how we're thinking about ovarian cancer. We're thinking about coming at those mechanisms of resistance. And I'm going to give you an example of one success in a moment. We're thinking about how we can target the proteins that coat the surface of ovarian cancer. Antibody drug conjugates are an example, and I've already given you several examples of those. We're thinking about how tumors regulate their growth and division. That's the cell cycle. And we're actually getting closer to having drugs that work and are tolerable, key point, to move into big phase 3 studies. And as I've mentioned several times, we're getting better at defining how we individualize a certain therapy for each individual patient, not just based on how long it's been since last platinum. That's a dinosaur way of thinking about this. There's a lot of factors that have to come into play, and that's really going to optimize outcomes for patients moving forward. So let me just give you a few examples as I finish up. Understanding mechanism of resistance. And I know there's a lot of MD Anderson scientists on this call right now. They really led the basic science behind understanding glucocorticoid receptors and their importance in creating resistance mechanisms for tumors, especially ovarian cancer, in relation to certain chemotherapies. This has been known for years. We finally have a drug, relacorilant, which just had its phase 3 study announced. And I'm showing you the Kaplan-Meier curves where the addition of relacorilant to a taxane improved or decreased the risk of progression by 30%. And looks like, we can't say it's definitive yet, but looks like it's also decreased the risk of death. So improved overall survival. We're still waiting for that final readout, and this is before the FDA now. So when we think about traditional chemotherapy and taxanes, we're entering an era of combinations where you could get weekly paclitaxel Avastin, perhaps Abraxane relacorilant in the near future, and other opportunities, which may move it into a preferred kind of regimen opportunity. I'm going to skip through this in the interest of time. The next example, I've been talking about antibody drug conjugates. This is the first one approved in ovarian cancer. Mirvetuximab soravtansine targets folate receptor alpha. It's approved for tumors that are folate receptor alpha high, which is about 40% of tumors. And is given as a single agent. This is the first study to improve both progression-free and overall survival ever in platinum-resistant ovarian cancer, and sets the bar for us that we can do this now. We can hit overall survival and improve it with targeted therapies and use of biomarkers. There are more coming. This is Rina-S, Rinatabart, another folate receptor alpha targeting antibody drug conjugate with a different chemo on its tail. So it's an entirely different molecule. This is in a phase 3 study in platinum-resistant ovarian cancer. And it raises the question that I think we're going to have to face pretty soon of how I'm going to sequence this for you. Because maybe I'm going to use mirvetuximab first and kill the folate receptor alpha high cells, because that's where it works. Rina-S appears to work irrespective of biomarker. So then I use that, and it probably works pretty well. And I get some additional time. What if I reverse them and I use RENAS first? Maybe it works a little better because it works on all the levels of folate. But if I come in with mirvetuximab second and it doesn't work as well in folate low, maybe 1 + 2 doesn't equal as much as the first sequencing. I don't know this is true at all. I'm just thinking about it. We're going to have to figure out sequencing of these drugs so we can line them up correctly for you to really make sure we're optimizing survival and quality of life. Or we switch to a completely different target. This is raludotatug deruxtecan, which is in a phase 3 study. You all will see the phase 2 data for this at ESMO in a few weeks. So this is exciting. We have bispecific antibodies, novel immunotherapy coming. I think this is a little farther behind antibody-drug conjugates, but may have a big role as a maintenance. These are very well tolerated medicines, not chemotherapy. They don't shrink tumors well, but they hold things stable. So you could imagine in a maintenance setting, these might be highly effective. And so I'm excited to see where these end up. And then I mentioned cell cycle, which is something many of us physicians on the call have been working on for a long time. WEE1 kinase inhibitors, ATR inhibitors, they work, but they've been kind of toxic, too toxic to move into big studies until the one on the right. And this is Insight's CDK2 inhibitor. This is data from ASCO, where they tested this in platinum-resistant ovarian cancer that was biomarker selected to be something called cyclin D1 overexpressing. So it's another protein target. And they had a response rate of 36%, notably with an incredibly clean toxicity profile. Like patients really felt pretty good on this, which makes me excited. And this is moving into registration enabling studies. And so I have my eye on this one for a lot of different settings. And then I mentioned this a couple times, got to move away from treating you all like dinosaurs. Like it's just madness to say greater than 6 months, less than 6 months. That's the end all be all of how I pick your therapies. It's so much more nuanced, and clinical trials moving forward will reflect that. So we can actually start individualizing in the way that we need to be doing. So in conclusion, I think my last point is my first point. One of our biggest advantages is to design studies that correctly characterize our patients by more than just this dichotomization of greater than, less than 6 months. That's going to transform how we study medications and move them into practice. I think our biggest win here right now, our biggest potential winner around antibody-drug conjugates. They are everywhere. They're moving into every line of therapy. I'm excited but worried. We'll talk about that. And then there's just this continued urgent need to understand the true biology of these tumors and where we fit in some of these other sorts of nuanced therapies to best take care of patients moving forward. And sequencing is going to become our friend. I say this all the time on stage. We've never worried about sequencing in the past because we did not have an armamentarium of drugs that worked well enough to matter. Ineffective drugs work no matter what order you put them in. But now that we have effective drugs, how we sequence them is going to be important. So we're going to have to really get on testing that so that we can advise on how we line these out. And with that, I thank you for your attention, and I will be around for questions. Thank you.